how do we go from this genetic information all the way to this protein that is made? So here's our cell, here's the nucleus. Inside, again, you have all that genetic material. And then from that information, okay, depending on the sequence of the genes the sequence of the bases that makes those different genes will result in the coding of a certain type of protein. So we know that the DNA controls function. It controls the production of proteins. For example, enzymes that are being made, antibodies that are being made. We already know what those are. Receptors that are found along cells that will accept neurotransmitters. So it's really, really important that we have the right sequencing for the right type of proteins that we need. Okay, so there is a gene for every single protein in your body. So again, here's your DNA. The length segments of length within that DNA shows all the different genes that are on there. Now, we make a little copy of that, which is known as the messenger RNA. This happens inside the nucleus. That gets passed out into the cytoplasm of the cell, where it then will code for certain amino acids that come together to make a certain type of protein. Because remember that, right? Remember that proteins are made out of the subunits, which are amino acids. So humans have roughly 20,000 to 25,000 different genes in them, but we're not expressing them all at the same time. We're not always making all these different proteins. It depends what our bodies actually need. All right, so I mentioned earlier that there are different types of proteins that are made from our genes. So for example, hemoglobin shape, do you remember hemoglobins, those proteins that are found in red blood cells? They are controlled by a sequence of bases that make up a certain type of gene. So this could be really good for you, but also really bad for you, because if the sequence of the gene results in a clump-shaped hemoglobin, that leads to a type of red blood cell called sickle cells, which is not good for you. And if you get both copies from your parents to have sickle cell, it can lead to death, right? It usually does lead to death. But for most of us, right, especially in this part of the world, we have normal hemoglobin, so normal shaped, meaning that the sequence of the sequencing of the amino acids we have resulted in a certain type of protein that is, in this case, a normal shaped hemoglobin. Okay, so again, normal shaped hemoglobins result in our normal shaped red blood cells. Clumped hemoglobin results in something called sickle cell. You can tell that these ones then are not going to be very good at carrying oxygen. Okay, just to look at this again, if we go inside that DNA, okay, we're pulling up the DNA and we're making a copy of an mRNA from it, and then that's what eventually leads to that protein being made. So here's the thing. Remember that in DNA, you have apples and trees, AT, cars and garages, CG. But when you're looking at an RNA molecule, it's only half the strand of that DNA. And you'll also notice that it doesn't have any T, it doesn't have thymine. So instead of a T, they have something called a U instead. So that means that instead of adenine A always being paired up with T for thymine, for RNA, you will always replace the T with the U, okay, and that is for uracil. This will make a lot more sense once we start looking at how to make proteins. Okay, so I know this looks really, really scary, but let's try to simplify this. Over here is the bottom part of, or whatever, the top part, a part of the nucleus of a cell. So here's the nucleus region, here's the nuclear pores, and outside here is the cytoplasm. This hamburger structure here, the top bun and the bottom bun, that is a ribosome. Okay, remember that ribosomes, their job, their function is to make protein. So it's for protein synthesis. All right, inside here you have your DNA. Remember this long structure of DNA. And DNA has two sides of the strands. All these little color like pegs, like they look like teeth. Remember, those are your bases, right? So like ATG or ATGC. Okay, so what happens is in the middle, so some part of that DNA where there's a segment of a gene, okay, it unravels and it opens up. And by opening it up, it separates the pairing of those matching bases. So remember, A and T and C and G. So it separates it up. And what this allows is it allows for the free nucleotides that are floating inside that nucleus to come and pair up with one half of that DNA molecule. When this happens, you get a new structure that's made. So you can see it right here. Okay, there's one nucleotide, there's a free nucleotide coming in and pairing up. And what you're making here is an mRNA. So remember, being an RNA, when you have an A on the DNA strand, you can no longer pair it up with a T, but instead you replace it with a U. So even though in this diagram, you don't see the little teeth-like pegs coming out and shown here, like as you see here, they are there. And you can see it really clearly down in this image here. Okay, so step number 1, all the genetic information, the coding is always found in the nucleus, right, in that DNA. Okay, that's important to know that. Step number 2, an mRNA, okay, is made and it carries a copy of that particular gene, so this one right here, out into the cytoplasm. So it passes through the nuclear pore and comes out into the cytoplasm where it meets up with a ribosome, okay? Once it gets to the ribosome, it's step number 3, okay? Remember, all the mRNA has the pegs coming off of them. It passes through the ribosome. And when it does this, as it's passing through the ribosome with each of its own base, a different type of RNA called an nRNA that carries a certain type of amino acid, depending on the 3 base it has, it will bring over the amino acid and start linking them up together to make a protein structure. Well, first, remember, it makes a polypeptide, and then depending on the properties of those amino acids, will kind of bound together, or will form together to make that protein. Okay, so are we good so far? Hopefully that makes sense. Okay, so that's number 4. So again, that protein that is made is due to the specific sequence of those amino acids that come together. And that is due to the specific sequence of the mRNA bases. So for example, if you see here, the mRNA, it's AUG. AUG results in TAC because it's the opposite, well, not the opposite, the pairing complementary bases, and that will bring in this particular amino acid that will join in. Okay, and the next one gets joined in, and the next one gets joined in, and eventually you get that long polypeptide. And that's how you make a protein. Okay, this is just another way of looking at the image. Again, here's your DNA, it becomes unwound, the mRNA down here gets made, mRNA leaves, passes through the ribosome, that tRNA comes along, brings in a specific type of amino acid that joins up to make that long polypeptide, eventually making that protein. Okay, all right. So for the last step there, number 5, I want to be really clear on this. When you look at the that mRNA in its sequence, right? Depending on the sequence, it will result in a certain amino acid coming in and then eventually making that polypeptide. So for example, if you've got the sequence of AAA, so adenine, 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 that will go and result in a tRNA bringing in a lysine amino acid. At this point for you guys, you don't need to know the different types of amino acids. I want to put some context here so you understand that. Here's AGG, okay? That particular 3 codon of those bases result in something called arginine being made, okay, another type of amino acid. So if you see here, these are different types of amino acids that are brought over. So they're actual chemical structures. Here, we just simplify it by drawing a sphere. Okay, don't worry about this. This is just the different ways that the 3 different codons, like a basis, can come together to make different types of amino acid. So just ignore it.